Hey guys, so as we close in on this AP Bio exam, you may be starting to get a little nervous. Don't worry about it. It's just an exam. It's not that big a deal. 5 years from now, it won't make a difference. You won't even remember, and no one will care. There are definitely some strategies we can use on the AP free response that will help us get as many points as we possibly can, and we want to make sure we're using those. So I'm going to do this free response question, show you exactly what I would do, and you'll be able to use these tips to definitely improve your score. First thing you want to do is take a look at the other file that I sent you in this announcement that has a free response question. I want you to try that on your own, do your best with it, see what you can do, and also maybe time yourself to see how long it takes you. Write out actual answers. Before we kind of like made quick sketches of answers, this time we want to start writing actual answers, write sentences. So don't watch this video until you've done that. So at the beginning of the question, the first thing it says is read each question carefully. Write your response in the space provided for each part of the question. Answers must be written out in paragraph form. Outlines, bulleted lists, or diagrams alone are not acceptable and will not be scored. You're not going to be drawing a diagram, not in this year's test. But you want to be careful. You don't want to write outlines. You don't want to write lists. You want to answer questions with sentences, always sentences. And remember that every single point can be earned with only a single sentence. So you don't need to write too much. You want to answer as many questions as possible. There's a thing that AP said recently in a presentation that they did online that said that it's going to be possible to get a 5 without answering everything. And that tells me that they may be making these questions bigger than what the time allotted really allows for. So if we're going to be leaving something out, which is possible, not preferable, but it is possible, we want to make sure that the thing we're leaving out is the thing we were going to miss anyway, right? The hardest thing for us, right? So we want to make sure we prioritize. So let me give you a few tips. So there's a lot of words here. This is all the description stuff. I kind of got it all into one slide, so it got a little crowded. In an AP Bio free response question, you want to read everything, even if it's just a quick skim. You don't need to give it a thorough read right now. What you need is to be able to come back and find the places that answer each of these questions in a timely fashion. So it looks like at the beginning we're talking about electrons from oxidation of glucose, glycolysis. So this is all cellular respiration stuff. Electron carrying NAD plus, NADH. Remember, right, it carries 2 electrons because if you think about it, if you've got NAD plus and you've got H plus and you put those 2 things together, right, there's no electrons to put those together. Neither one of them has electrons. So it's going to need 2 electrons to form the bond between these 2 to make the NADH. Right, so those 2 electrons are taken from glycolysis or the Krebs cycle or pyruvate oxidation, and then they are used at the electron transport chain. That's the electrons that are passed to that first guy that makes it so that it gets the shape change so we can start doing that pumping protons thing and generate ATP that way. So during glycolysis, we make these guys, and then we have to get these guys back for more glycolysis, and normally we get that through the electron transport chain when we have oxygen available. If we don't have oxygen available, we do fermentation. Okay, that's all review of cellular respiration. There's nothing really new there. And hopefully you've looked at the cellular respiration again. If you haven't now, hopefully you have before you take the exam, right? Because we want to make sure you've gone over everything again. Go through all my old PowerPoints, get your notes out, read through those, make sure everything makes sense. If you have questions, please shoot me a question. I can answer the question in an email. I can make a video answering the question. More fuel for videos is always good. That's the thing that's really missing from this online education thing is that interaction. So I don't know where you guys are. So there's all this stuff about cellular respiration and NADH. Pretty straightforward as far as that goes, but that we know that's there. And now it says we've got this gene MTND5. It's a mitochondrial gene. So that's of note, right? That's interesting. It's not part of our normal chromosome. It's in the mitochondria. So this mitochondrial gene codes for a subunit of NADH dehydrogenase. The enzyme catalyzes the initial oxidation of its substrate. So it turns this guy into these guys. That's what this says in the electron transport chain in the mitochondria. Okay, so now we know where this is. It's that first guy that takes the electrons from the NADH. Mutation is associated with a rare genetic disorder that results in a buildup of lactic acid, which kind of makes sense when you think about it. Maybe you're not thinking that much at this point because we're just giving our first skim through, but it ought to be kind of clicking that if this guy doesn't work, we can't do electron transport chain. We're going to do fermentation instead. So if we are doing this with less efficiency, then we're going to have to do more of this. And hopefully that makes sense. The researcher hypothesizes that mutated has decreased activity but not completely non-functioning. That's not surprising. You probably wouldn't be alive if it was completely non-functioning. If you increase the amount of NADH, the activity will increase. To test the idea, here's some data, blah, blah, blah. All right, let's see what the questions are. Before I start writing any answers, I want to look at all the questions and I want to write down a couple of things about them. Since this test is going to be on your computer, have a piece of paper. You should actually have a stack of paper, scrap paper there available. But have a piece of paper and just, question 1, A, B, C, D, E, have it written there. So for this first question, describe the pattern of inheritance that is most likely associated with the mutation at MTND5. So pattern of inheritance, we already heard it was a mitochondrial gene. That's a pretty easy one, right? So you want to put on your notes a little smiley face on that one because that's not too bad, right? You're going to get it from mom because you get your mitochondria from mom. That's your pattern of inheritance, right? How's it going to show up? Explain why individuals are not typically heterozygous. That's not too bad either. Maybe kind of a meh face. Or maybe a smiley face if you're thinking, oh yeah, you can't be heterozygous for mitochondria. You only have one type of mitochondria. You don't have a pair of chromosomes. You don't get some from one parent, some from another. You only have one type. So maybe that's not too bad. And this looks like it's 2 points, right? Total. That's the other thing you should be writing down. So 2 things you're writing down when you go through this the first time. Smiley faces, meh faces, frowny faces, right? Based on how confident you are that you can answer these questions. And then the other thing you should be writing down is how many points do you think it's worth. So this looks like 2 questions, 2 points. Both of them not too bad. Let's go on to the next one. So looking at part B, identify a dependent variable measured in the research experiment. Dependent variable is easy. That's y-axis, right? That's the one that depends on what you manipulate. So it's the thing that you're going to be measuring how much it changed. So that should be easy. You've got 2 graphs. It's going to be one of the y-axes, right? So that's a smiley face. At least I hope it is. Identify one control that the researcher could use to improve the validity of the experiment. Control's not too bad either. That should be easy to find. We'll look at the data. Justify the researcher analyzing blood samples at many intermediate time points instead of only at the beginning and at the end of the 20-week period. So why is it better to look at lots of data points as opposed to just at the beginning and the end? Yeah, that's easy too, right? More data is more, right? That's good. And it looks like there's 3 questions there, 3 sentences. We got 3, and none of those look very hard. This is just kind of basic experimental design stuff. You're going to get questions like these. Absolutely. They've already said that there's going to be an experiment involved on the first question that you're going to have to do some evaluation on. These are the kinds of questions they're going to ask. Looking at part C, describe the relationship between the concentration of NAD plus in the blood and the concentration of lactic acid in the blood in the first 5 weeks of treatment with the vitamin. Describe the relationship. So that's this graph here. In the first 5 weeks, so that's going to here. I assume this is weeks on the x-axis. Yeah, it's time weeks. So what's going on right here is all it's asking. And that's not too bad. It's going up. So that doesn't look very hard. Based on figure 2, calculate the average rate of change in blood NAD plus concentrations from week 5 to week 17. So how did it change from week 5 to week 17? That doesn't look too bad either. We got a data point there that looks like about 50. We got a data point there that looks like about 300. That's not going to be hard to do. We got time. We got change. We can do a little math. That doesn't look hard either. Math. Part D, the researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells. Predict the effect of the MTND5 mutation on the rate of oxygen consumption. All right, so this one might be a little harder, but it's going to take some time, right? And you might even put a little clock on there, something that tells you this one's you're going to have to think about for a bit. Justify your prediction. So that's going to be explaining that. Oxygen consumption, electron transport chain. There's definitely a connection there, but it's going to take some time. So we want to save this one till later. The researcher had a hypothesis that the addition of the vitamin that is similar in structure to NADH would increase the activity of the mutated. Okay, so here we're talking about enzyme function and adding more substrate. And then we have to explain that. So these are definitely these are harder. And you might even have a frowny face on this page. So we want to hold off on this one. And it does look like they're saying 3 things here, so that's 3 points also. Going back to part C here, I forgot to put how many points this is, but it looked like it was 2, right? So we've got 2 points, 3 points, 2 points, 3 points, a total of 10 points. This question definitely looks to be the hardest of all of them. Let's figure out where we should start, though. Now, you may be thinking, Mr. Powell, shouldn't we be writing during this time? No, we shouldn't. I mean, we're making some notes, but we need to be making sure we're doing this in the right order, and that's our first priority. That's the thing we want to do first. So would you take as long as I am doing this? Hopefully not, because you're not explaining it to a camera, but you definitely want to take this time. You can start on any of these. I would probably start here on part C, just because it looks super easy. Describe the relationship between the concentration of NAD plus in the blood and the concentration of lactic acid in the blood in the first 5 weeks of treatment with the vitamin. Again, going back to that graph, first 5 weeks, it's going up. The concentration of NAD plus goes up dramatically. So to answer this question, the concentration of NAD plus in the blood, oh, and it's also asking about the lactic acid. So I don't think that did go up. I think that went down. First 5 weeks, yeah, it goes down. So all we have to do is say that in the first 5 weeks of the treatment with the vitamin, the NAD plus in the blood went up and the concentration of lactic acid went down. So you could just say the NAD plus went up while the lactic acid went down. Based on figure 2, calculate the average rate of change in blood NAD plus concentrations from week 5 to week 17. So week 5, we have, again, it looks like about 300. And week 17, it looks like it's about 50. So that's 250 in 12 weeks. Right, 17 minus 5 is 12 weeks. So calculate the average rate of change in blood NAD. So that's just 250 divided by 12. And that equals, I got 20.83. So, sig figs, that'd be more like 21. I would say something like from week 5 to week 17, the NAD plus concentration in the blood went down by about 21 micromoles per liter, because that's what it is on the graph. Micromoles per liter per week. Okay, so those 2 points are done. So X off C, we're done with that. B, identify a dependent variable. Which one's easier? Let's go with this one. But we want to always pick the next easiest one, right? Always, what's the easiest one next? What's the one that your brain says, I can answer that right now? Identify a dependent variable measured from the researcher's experiment. So dependent variables are on the y-axis. So let's just say concentration of NAD+. So you would write a sentence saying a dependent variable measured in the experiment is the concentration of NAD+ in the blood. That's all there is to it. Or you could say a concentration of lactic acid in the blood. Either one of those is perfectly acceptable. 1 point for either one of those. Identify one control that the researcher could use to improve the validity of the experiment. So let's look at this again. So the test said treated a group of individuals with the disorder with a vitamin that's similar, and then watch what happens. So we don't have a control group at all. There's nobody. We want a group of individuals with the disorder that didn't get the vitamin, right? That would be a control. That would be perfect. It would be all of the factors except for the vitamin. That's what we want. Control is pretty easy to find. Like I said, usually you just have somebody who is standing there and not getting the experimental treatment. In this case, you would want a group of people who are not getting the experimental treatment. Being humans, it's good to give them a pill to make them think that they are getting something, some kind of placebo. If you're giving a pill to these people, then the control group needs to get a pill also. So you could write a sentence like, to control the experimenter should have a group of people with the disorder who are taking a placebo and measuring their blood levels. Right? Control gives you something to compare against. So that's what you're looking for. What am I going to compare it to? What are they going to be like if I don't do the experimental condition? That's what a control tells you. Okay, so that's when it asks you for a control, and they do this all the time. Give me a control. Right? That's what you want to do. Show that you're doing everything except for the experimental factor with another group. Justify the researcher analyzing blood samples at many intermediate time points instead of only at the beginning and the end of the 20-week period. So beginning and end, if you look at these graphs, right, if you had just gone beginning and end, you wouldn't have seen much change. You would have seen a big change in the concentration of lactic acid, but the change in NADH, you would have missed this whole spike. Also, by gathering data all along, you see that most of this drop happened very early in the first 5 weeks, and then it barely went down at all after that. So you got a lot more information with more data. So when we say justify, we could just simply say that, having more data allows the researcher to explain what happens a little more effectively, right? More data is always helpful for better conclusions. More data, more confidence in our results. Part A, describe the pattern of inheritance, right? So this is a mitochondrial gene, so you don't have to just say something like mitochondrial genes are passed on from the mothers, so they would always get it from their mom. Explain why individuals are not typically heterozygous, right? Mitochondrial genes, you don't get them from both parents, you don't have pairs of them, you don't have pairs of the chromosomes, so you can't really have two different versions like the nuclear chromosomes have. So really all you have to say for this is you don't usually have pairs of chromosomes for mitochondrial genes. All right, so now let's look at that hard one. We've answered all three of the easy ones. The researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells. Predict the effect of that mutation on the rate of oxygen consumption in the brain cells. Okay, so that's our first question. We need to predict, and that would probably be bold typed in the actual test. The researcher had hypothesized that the addition of the vitamin is similar to the structure of NADH would increase the activity of mutated NADH dehydrogenase enzyme in individuals with the disorder. So if it works well enough to mimic NADH, it's going to make that enzyme do its thing if it's that similar. So that's his hypothesis. That seems sound to me. Explain how the vitamin most likely increased the activity of the enzyme. So oxygen consumption, right? We use oxygen, if you remember your electron transport chain, and that's what this question's asking you. You use oxygen as that final electron acceptor. As we pass the electrons through the electron transport chain, the last one we give it to is oxygen. And we said before, if you don't have oxygen, then that all stops because you can't pass electrons to something that still has electrons. So if you're not passing electrons to oxygen, the entire electron transport chain stops. So what's going to happen to the amount of oxygen consumption? It's going to go down because we're not doing the electron transport chain as much. We're not going to be using as much oxygen. So like I said, you get 2 points for this first part. First, predict the effect. That's going to be less oxygen consumption. And justify your prediction because oxygen is the final electron acceptor in the electron transport chain. And if we're reducing our activity on the electron transport chain because of this mutation, then we're going to have a reduction in the use of oxygen. All right, so for the third point, explain how the vitamin most likely increased the activity of the enzyme. So it binds the active site of the enzyme. It is similar enough to NADH that it can bind the active site of the enzyme and make it work. And that's really all you have to say for this one. It's similar enough in shape to bind the active site. That's all you need to say. All right, so when we take an AP free response question, the first thing we want to do is skim through the entire question, all the information they give us. We want to go through all the questions and identify how many points they're worth and how challenging you think they are. And then you want to make sure that you complete them in the order from easiest to most challenging. Right? You don't want to leave any points out there that are easy to get. You want to make sure you've already done those. Remember, every point can be earned with a single sentence. Don't go nuts. Find the points, give a sentence, right? For each point, give a sentence. Probably most important thing, be well rested, man. This is not about what's in your brain. It's about using your brain. And a well-rested brain is much more useful. So sleep. I know that some of you guys, since this quarantine, it's not really a quarantine, has begun, sleeping has not been something that's regular. You need to get regular for these AP exams. You need to make sure you're going to bed at a reasonable time, waking up at a reasonable time consistently. Okay? It's the irregular hours that are going to make you sleepy and make your brain foggy and are going to make you tank it. Being awake and well rested is going to make a huge difference on this test. Okay? You're going to have to think on your toes. And you're going to have to do it in a short time period. So you need that active brain, that really working brain. You need your best brain. It doesn't need to be full of stuff. It needs to be working well. Love you guys. Hang in there.